Welcome, and thank you for joining us. Our next presenter this afternoon is Olivia Veach. Dr. Veach is an assistant professor in the Department of Psychiatry and Behavioral Sciences at the University of Kansas Medical Center. She has a PhD in human genetics and credentials in molecular biosciences, bioengineering, and translational bioinformatics. Dr. Veach will report on the impact of ATP-sensitive potassium channel gene variants on baseline insulin and hyperphagia in Prader-Willi syndrome. Please welcome Dr. Veach.
All right, so hopefully everyone can see my slides. So I just want to start by thanking the organizers for giving me the opportunity to speak today about this work. So this is a genetic study that we conducted looking at how individual variation may impact baseline characteristics in patients with Prader-Willi syndrome that could influence how they respond to treatment. So I'm just going to start by giving a little background on how the compound of interest works. So this compound treats hyperphagia, and why we thought that variation impacting the evaluated genes may influence characteristics that could affect a patient's response to this treatment. So the hyperphagia signal in Prader-Willi syndrome is likely occurring when a group of neurons that includes neuropeptide Y, a GUDI-related protein, and gamma aminobutyric acid are dysregulated. This seems to be associated with deletion or a disruption of SNORD116, which is encoded in the Prader-Willi syndrome critical region. And these neurons, when they're dysregulated, result in elevated synthesis and secretion of neuropeptide Y and a GUDI-related protein. So the leptin hormone works endogenously to regulate these neurons, and it does this by activating these adenosine triphosphate sensitive potassium channels. This then hyperpolarizes the resting membrane potential and limits the release of neuropeptide Y, and this ultimately helps to maintain healthy insulin levels in the blood. So exogenously administered diazoxide choline has been observed in vivo in animal models to more extensively hyperpolarize the resting energy potential of neuropeptide Y, a GUDI-related protein, and GABA neurons than does leptin. So diazoxide also is known to cross the blood-brain barrier. So therefore, orally administered diazoxide choline can effectively agonize these ATP-sensitive potassium channels, and particularly in the neurons of interest in the hypothalamus. And this works to supplement the effects of leptin, blunt the hyperphagia signal. It also serves to hyperpolarize resting energy membrane potential of pancreatic beta cells, effectively reducing excess insulin secretion. So there are actually 4 genes of interest that encode subunits of these ATP-sensitive potassium channels. So this includes 2 genes on chromosome 11, ABCC8, which encodes SIR1, which is one of the subunits, and then KCNJ11, which encodes KER 6.2. And then there's 2 genes encoded on chromosome 12, ABCC9, which also is encoding SIR complexes, and then KCNJ8, which encodes KER 6.1. So our hypothesis was that variation affecting the genes that encode the subunits of the ATP-sensitive potassium channels could influence baseline characteristics, specifically insulin levels and the hyperphagia signal in individuals with Prader-Willi syndrome, and that this may actually impact how the patient responds to treatment with orally administered controlled released diazoxide choline. So we collected data from 118 individuals with Prader-Willi syndrome who were included in a clinical trial for this compound. And I just want to note that our patients were mostly reported to have white ancestry, and a little over half of the data set were reported female. So again, our outcomes of interest were baseline insulin, and we looked at that as a quantitative measure, and then as well as hyperphagia scores. We looked at both of these as quantitative continuous outcomes, but we also looked at them based on a split at the 75th percentile of the distribution of these measures in this data set to target the extremes. And I'm just showing you here what those 75th percentile measurements were. So we initially evaluated associations between insulin and hyperphagia baseline measurements and genotypes at 18 different single nucleotide variants. These were located near coding regions for genes of interest, and it included 7 variants on chromosome 11. One was downstream of KCNJ11, and 6 were located within the coding region of the ABCC8 gene. We also looked at 11 variants that were located within the coding region of ABCC9 and upstream of KCNJ8. And I just want to note too that we defined the effect allele for these associations. This was defined as the minor allele, and that was based on populations of European ancestry, since the majority of the patients included in our data set were white. So there were 3 variants that were correlated with the continuous outcome of baseline insulin. One of those variants, interestingly, it was downstream of KCNJ11, and it's actually located in an area of the genome that's predicted to house regulatory elements, notably enhancers or potentially transcription factor binding sites. And genotypes at this variant were actually associated with expression of KCNJ11 in a number of human tissues included in the GTEx portal. And I just briefly want to let you know what the GTEx portal is in case you don't know what it is. It's a publicly available resource of RNA-seq data. And these data are generated from various human tissues. They're all obtained from postmortem biospecimens from typically developing individuals. But this actually allows us to identify single nucleotide variants that are associated with gene expression in these various human tissues. And these variants are called expression quantitative trait loci, or eQTLs. So most notably, the insulin-associated allele in this variant, the T allele, is associated with a slight decrease in the expression of KCNJ11 in thyroid. And evidence indicates that thyroid dysfunction can have an impact on food drive and metabolism. We also looked at groups of variants that were co-segregating and observed a group of variants within the coding region of ABCC9 that were in strong linkage disequilibrium, or LD. And this indicates that they're being inherited as a haplotype. We observed that being heterozygous for the less frequent alleles in Europeans at this haplotype was associated with having a baseline insulin level that was greater than or equal to what the measurement was that reflected the upper 75th percentile of the distribution of insulin across the data set, or 12. And we wanted to look further at these functional variants that were associated with expression of our genes of interest. And so we went into the GTEx portal and looked for any variant that had not yet been tested, not the original 18, that were associated with expression of any of the 4 genes in any human tissue. And we did observe that there were 83 of these predicted expression quantitative trait loci outside of the coding regions for this genes. This included a group of 8 variants that were all in strong linkage disequilibrium. They were downstream of the coding regions for KCNJ11 and ABCC8 on chromosome 11. They were located, all of them, in a genomic region with a number of predicted regulatory elements, mostly enhancers are predicted here. And they were associated with expression of ABCC8 in a number of tissues. They were also associated with baseline insulin levels in the Prader-Willi syndrome patients. And I just want to note here that this haplotype of insulin-associated eQTLs was correlated specifically with expression of ABCC8 in the cerebellum. And the effects of the insulin-associated alleles were an increase in overall ABCC8 cerebellar expression. So increased baseline insulin levels in the blood of patients with Prader-Willi syndrome, and increased expression of the ABCC8 gene in the cerebellum from typically developing individuals. And also notably, the cerebellum is where this gene is observed to have the highest level of brain expression in human tissue. And the cerebellar region is also where the motor nucleus of vagus resides. And this is a key target for ATP-sensitive potassium channel activation in the treatment of hyperphagic obesity. I'd also like to point out, even though there's less expression here, that ABCC8 expression is detected in the hypothalamus, and also noting that these variants were not associated with expression of the gene in this region, but the hypothalamus, as I indicated in the introduction, is an area of particular interest to agonize for these ATP-sensitive potassium channels in these neurons. So I just want to summarize now so we can move on to questions, but, and I know I went a little fast, I talk always fast and try to slow down and it never works. But in summary, these single nucleotide variants that were associated with expression of KCNJ11 and ABCC8 were also observed to be associated with baseline insulin. This may influence how a patient responds to diazoxide choline controlled release. Specifically noting that a variant in an enhancer element near KCNJ or KCNJ11 was correlated with insulin levels as baseline, as well as expression of the gene in the thyroid. There was also a haplotype of 8 SNPs that were located in predicted enhancer elements that were associated with increased baseline insulin in the blood, as well as higher expression of the ABCC8 gene in the cerebellum. And so this actually indicates a possible relationship between levels of insulin in the blood and expression of this gene in the cerebellum. And so ultimately, I do want to point out, though, that these results do need to be confirmed in independent data sets. So our sample size wasn't super small. We had over 100 patients, but it still would be good to replicate these findings in independent data sets. And the other thing I'll note is, as I try to point out throughout the talk, that these analyses were really focused on individuals of European ancestry. And so they may not generalize to populations with different ancestral backgrounds. So just something to keep in mind moving forward. So I want to take this time to thank my co-authors. I specifically want to thank Dr. Manzardo, who generated a lot of these results that I presented today. I want to thank all of the families that participated in the study and allowed us to do research on their DNA and their data, and also thank my funding sources. And with that, I will take questions.
Hello, wonderful. Thanks so much. You always have a wonderful presentation and terrific slides and a way of making things seem very clear. I have to give full disclosure, as she mentioned, I am a co-author on this, on this report, and we did work on this together. So, so I'll throw some of this to Dr. Poggi as well. They had some basic questions about the age range, which I know we talked about.
Yeah, we did talk about that, and we are not confident on what the age range was for these patients, and that's just one of the caveats of working with data sets that were collected by someone else. And so if I do remember some of the work that has preceded this in the clinical trials, it was a pretty broad age range that's being looked at for this compound. And
Right.
so I apologize that I can't answer that question accurately. So
I'm partially to blame that too. We did not have age in that sample. So now insulin observance was prominent in the study. What about patients that are not diabetic and therefore do not need to watch their insulin level?
Ooh, that's a good point. Yeah. So I am not sure if patients with diabetes were excluded from this data set, but I don't think that there were patients with diabetes based on just the distribution of insulin, and maybe Dr. Manzardo, you could speak to that since you also know those data. But I think that this isn't necessarily about diabetes or not diabetes. It's about insulin regulation, and that points to the function of these potassium channels, these ATP sensitive potassium channels that we're trying to target with the compound. So if you're trying to reduce insulin by targeting these channels, it's possible that in patients who have higher levels of insulin at baseline, even if they're not diabetic, that their response to the treatment could differ. And so that could impact how your drug trial would go. You may see that you want to try to identify patients who don't have these variants that are influencing their baseline insulin if you're looking for something that's going to reduce insulin levels. So that would be just one example of why that would matter.
Right. And this study did not investigate whether or not the DCCR response was affected by these variants.
So I'm not reporting any of those results. We are looking into that. And there's a team of investigators who are working on that. And so hopefully at some point in the near future, we can also report on what might be impacting the response to the compound.
So what about these variants being associated with sleep apnea? Do you remember the data literature on that?
Oh, I do not. So these variants are associated with sleep apnea, or have been?
They wondered if we did not find that.
Yeah, we didn't look for that, and it's actually it's a little ironic that someone asked that, because I've worked on sleep apnea, albeit not necessarily have a lot of results yet in the Prader-Willi syndrome population. So I did not make that connection, but I will definitely look into that. That's a really good question. Yeah.
Yeah, I hadn't thought of that. The idea that these variants might have some impact on sleep, and that was not something that was initially looked at. So I think those are the main questions. Dr. Koji, do you have anything you want to come in and ask?
I just want to, congratulate Dr. Veach on this great work. It was very, very interesting. I don't think I have any particular questions, however.
All right. Well, I think that we can wrap up a little bit early. Do we need to put up our closing slide? And we would ask folks to be sure to visit the vendors when you get an opportunity. And don't forget to fill out your surveys. Thank you so much, Olivia. Wonderful presentation.
Thank you. Thanks.